been following along, we've had 3 seminars in a row featuring cardiac and vascular research. And this is our 8th year in a row in doing that. And we invite internationally recognized experts in development, physiology, and disease in the cardiovascular system. You may have heard that tomorrow, February 22nd, is now recognized as Heart Valve Awareness Day. And so fittingly, our speaker this morning has contributed greatly to our knowledge on heart valve development and mechanisms of disease. So Dr. Joy Lincoln attained her PhD in molecular biology from the University of Durham in the United Kingdom. And then she moved to the United States to pursue postdoctoral training at Cincinnati Children's Hospital in heart valve development with Dr. Catherine Yutzi, who was a previous Heart Month speaker just a few years ago. After her postdoctoral training, Dr. Lincoln began her faculty position, first faculty position, just down the street at the University of Miami. And she was there for a few years, but then decided to give up the warmth and went back to Ohio, where she joined the faculty of Nationwide Children's Hospital and Ohio State University. And then just a few years ago, she was recruited to the Medical College of Wisconsin, so Ohio wasn't quite old enough. Went a little farther north, where she serves as a professor of pediatrics. She holds the Peter Summerhouse Endowed Chair in Cardiac Quality Outcomes and Research. She leads the Cardiovascular Research Program within the Herman Heart Institute, and she was recently appointed the Associate Chief Scientific Officer for Children's Research Institute at Children's Wisconsin. So in addition to being a well-recognized leader in heart valve research, Dr. Lincoln's passionate about mentoring young researchers. And also, I thought this was interesting, while doing a little research on my own material to introduce her, I learned from her website that she can deadlift 300 pounds. So I'm not just saying this because she can probably beat me up, but we are super excited to have her here today. And welcome, Dr. Lincoln.
Thank you, Josh, and thanks for the warm welcome. Literally warm welcome. I left -5 degrees Fahrenheit yesterday morning. In fact, I missed my connection, so we had to de-ice the plane. We're stuck at Chicago for 90 minutes, just de-icing the plane. Well, please, I know this is not a pure cardiovascular audience, so this is very informal. Please interrupt me. I have an accent as well, so if there's things I say that you don't understand, please interrupt me. So for those that don't know, as Josh says, I'm actually in Milwaukee right now. And there's some beautiful images there of the lake. We are very famous for our cheese curds and beer. So that's probably one of the main reasons why I moved there, was for cheese and beer. I mean, there's not much wrong with that. And then this was literally last week. So we had 1.5 feet of snow last week, and I say it's -5 degrees. But the really cool thing about our campus is that we have both the medical college, so we don't have any undergrad. It's all medical school and graduate school, attached to the Children's Hospital and the Adult Hospital. So on this campus here, we have bridges. We don't have to go outside in the winter. But I can then walk across the Children's Hospital, the Adult Hospital, the medical school, and have all child health and pediatric research under one roof. So it's a really convenient campus to create collaborations. So that's a little bit about where I am. So my favorite organ is the heart, and as Josh said, the valves are my favorite part of the heart as well. So what you're looking at here is the heart that beats 100,000 times a day. And the only reason the blood goes in the one direction is because of the heart valves. So there are 4 sets of heart valves. What you're looking at here is the pulmonic and the aortic. And then when we cross-section through the heart in a moment, you'll see the, you'll see the tricuspid and the mitral valves. And you can see they have those stringy chordae tendineae. And then we looked at the top there, you can see the aortic valve opening and closing, and they don't have those stringy chordae. So what I'm going to talk about today is really understanding the mechanisms of how in some babies they are born with structural malformations of their aortic valve and the complications of that moving forward as well. So again, this is what we're looking at here. This is the aortic valve on this side here. And if we look down, you'll see there are 3 cusps, so 3 flaps that open up and down. And if we took a really simplistic view of that on the left side, we call them the left, the right, and the non-coronary. So the coronary is the blood flow that goes across the valves. And you can see the left and the right there have these little coronary vessels that feed on, but the one at the bottom, the NC, the non-coronary, doesn't have a blood supply that goes across. So on the right side, you'll see here, this is the way the blood flow goes through. So now what you're looking at is the aortic valve cusps opening and the blood flow going through. So really there is a very simplistic, let's bring this to go here. The blood flow is going through in a laminar direction, so when the valve opens, the blood flow goes straight through the valve, and then we have this oscillatory or disturbed flow that goes when the valves close. So constantly in a healthy human, as they open, the blood flow goes from the ventricle straight through the aorta, and then as they close, we have this oscillatory or circulatory flow. So again, I think very simplistically, the clinicians, if there are any in the room, will cringe at this, but I think there are two major problems with the valves. Either they don't open enough, or they open too much, right? So in a normal situation, you want the valve to open. So here is where the blood flow will be coming through the screen towards you. And then in diastole, when they close, you have a really tight junction here, and the blood can't go backwards. So it's constantly going in one direction. So in some cases, there is a failure to open the valve fully. So usually we have this much space. And then some diseases, the valve cusp gets very stiff and it can't open fully. So if you think about a hose pipe, when you squeeze that and you kind of narrow the opening of the hose pipe, the water comes out really fast. So as I think of it, when my daughter squeezes the ketchup bottle, you know, it squeezes it out. So you're trying to push the same volume of blood through a much smaller hole. So the pressure behind the valve builds up so much that the heart has to pump even harder. And so this is a condition called stenosis. The other way is that the valve doesn't shut properly. So again, a really tight close here, but sometimes it doesn't close. And so what happens here is the blood goes backwards and forwards, and you don't want that to happen either. So today's talk's really focused on the stenosis, or the narrowing, the hose pipe situation of the valve. And so aortic valve stenosis is actually quite common. So 2.5% of the population has heart valve disease in general. There are deaths every year due to that. And then there are some babies pretty commonly born with a congenital malformation. So basically the babies are born with either a structural defect, or there's a structural change in the material properties of their cusp, which doesn't allow their cusps to open properly. And again, the cost for this on an annual basis is over $10 billion. So thinking about ways in which the valve might not open fully, there are three simplistic ways of looking at this. One is calcified. So this individual has 3 cusps, so a normal structured valve, but later in life, for whatever reason, they get these bone nodules form. And you can imagine that if you have a very compliant valve that opens and closes, if that material turns to bone, it becomes very stiff. And so that cusp now can no longer open. And so that's why you would get stenosis. The other way is just dysplastic, which honestly for clinicians, this is just anything they don't really know what the answer is to. Oh, it's dysplastic, which just means there's a structural change. So again, this individual was born with 3 cusps, so a tricuspid aortic valve, but their valves got very thick and fibrous. And so again, if you think about something being very fibrous, it becomes very stiff. So that valve can't open fully. And the other way is that some kids are born with only 2 versus 3 cusps. So here you can see there's just 1 cusp here, and there's 1 cusp there. And we're really going to talk about this bicuspid aortic valve disease today. So bicuspid aortic valve disease affects about 2% of the population. So 2 in 100 babies are born with this condition. They're born with it, it's congenital, it's inherited by genetics in about 25%. There is a male dominance. And the really interesting thing to me is that while these kids are born with these 2 cusps rather than 3, for some reason, these young adults develop calcification much, much earlier in life. So for someone with with 3 leaflets of their 3 leaves of their aortic valve, they would go through life, and if you were to develop calcification, you'd probably get that at about age 70. And for some reason, though, if you're born with 2 cusps rather than 3, you develop it at about age 35. So this process of calcification is very much accelerated. And the problem here is that these kids have not only 2 cusps, so it's already narrow, but then they get calcification of those cusps, so you get like a double hit. And so the impact of this on these kids' lives is actually very significant. And so my lab for many years has had many questions, but I'm very interested in these questions. For example, why are people born with 3 cusps on their valve, but later in life develop this degenerative calcific aortic stenosis? Why are some kids born with a normal valve, but they develop this really thickening later? and then why are babies born with these two cusps but develop this premature calcification. And so today's talk is focused on a project we've been looking at, really trying to understand these parts of why we go from a bicuspid to this premature calcification of the bicuspid aortic valve. So for you to understand what I'm going to tell you, you need to understand what the normal valve looks like. So what we're looking at here are 2 of the 3 normal aortic valve cusps, and we break them down into regions. And so as you think about the cusps opening, the very top of that is called the tip region, this is called the belly region, and this is the attachment region. So the attachment region is where the cusp joins the aorta, and it kind of has an anchor point. Okay, so as you open and close, this part here is the anchor point or the attachment. So if we were to do some histology of the aortic valve, I would show you that inside the tissue there are a layer of endothelial cells. So the endothelial cells line the complete surface of the aortic valve. So if you can imagine the blood is coming through here, these cells can feel the shear stress induced by the flow. So they're very mechanosensitive. They are also signaling hotspots. They're like this black hole. They hold all the magic signals, I feel. So any molecular pathways seem to be stimulated to come from those valve endothelial cells. So they line the surface, and then inside the valve are these interstitial cells, and these are like fibroblasts. And really their role is to secrete the extracellular matrix, and that includes a layer of elastin, and a layer of proteoglycans, and a layer of collagen. And so what happens is we call the layer of elastin is ventricularis. So as the blood comes through and the valve cusp opens, the elastin layer allows that to spring open, okay? So each layer, I think of it as like ice cream layers, right? So if you have each layer of the elastin allows the valve to spring open, the proteoglycans allow that compressibility. So when you have a force of blood flow hitting it, it kind of absorbs that, a little bit like cartilage in your leg. So as you put your leg down, it absorbs the power that's going through. And the fibrosa is made of collagen, and so that allows for tensile strength. So these valve interstitial cells, these orange cells, their job is just to spit out matrix and then degrade it in a physiological turnover. And so really in disease, the real problem is that the matrix gets disorganized. So if you have too much elastin, they're too springy. If you have too much collagen, they're too stiff. And so it's a very, very fine homeostasis of that extracellular matrix that's important. That's regulated by how the valve interstitial cells behave. So now if we, again, go back to the mechanical stress that these cells see, this is one cusp, you've got the valve endothelial cells on the outside. They feel many different stresses and they feel very many different strains. So they constantly have this wall shear stress being exposed by the blood flow, but they also have this compressive and tensile stress as well. So these endothelial cells sense this, and they will signal to the interstitial cells and say, hey, we need more collagen laid down, or hey, stop producing elastin, we've got too much. And so we have this kind of symbiotic relationship between these two cell types. So I feel that in healthy valves, we have this layer of endothelial cells on the outside, and they sense this shear stress. They signal to these interstitial cells and say, hey, just keep things going, right? We're a healthy valve, nothing needs to change, and they keep this in the right order. But then in disease, for some reason, some of these endothelial cells are damaged, or they're injured, or they're dysfunctional, and so there's a turbulence in blood flow, and then this whole regulation to the interstitial cells is disrupted. They start sending too much message out, or not enough message. And as a response to that, in the case of calcification, these interstitial cells then actually turn from this kind of fibroblast cell, and they start becoming an osteocell, like a bone cell. And so as a result of that, they lay down this calcified matrix. We don't really understand anything on the right pathway, right? We don't understand too much about how this happens in disease. And so my lab has been very interested in linking the mechanical indices and how that changes to the molecular signaling that might drive these cells to become a bone cell. So bicuspid aortic valve disease comes in several flavors. The normal is at the top, so we have the left coronary cusp, the right, and then the non-coronary, which we talked about. And when it's open, you get this beautiful opening and the blood can flow through. However, patients can be born with 2 instead of 3. There's a classic type 0, where basically they get born with 2 cusps. But then there are types where these cusps fuse. So the left and right can be fused together, the left and the non-coronary can be fused, or the right and the non-coronary can be fused. And again, 50% of patients who have any type of this bicuspid valve disease develop this premature calcification. And so as a result of that, in the normal case, you have this blood flow that goes through, but you can see here there's much more narrowing. So again, we're trying to get that same volume of blood flow through a space that's much, much smaller. And so the blood flow ends up getting skewed because the valve can't open, and those endothelial cells sense a difference. They sense that there's an increase in the velocity of the blood flow and the direction of the blood flow. So I'm trying to understand what are the consequences of those cells seeing a different biomechanical index in the presence of the bicuspid anatomy. So this is all work done by one of my graduate students who's getting ready to graduate and go to medical school. But we took our patient population and who have been diagnosed with fusion between the right and the non-coronary cusp, so a type 1 BAV. You don't really need to know that, but there's narrowing. And we try to understand what does this patient population do. So what we're looking at here, on the axis here is the peak gradient, and that's a measure of the opening of the valve. So the higher the peak gradient, the narrower the opening, which means they have more stenosis. So this is a real, these kids are more sick, right? You don't want that peak gradient. Our peak gradient should be between like 10, right? But some of these kids, the pressure going through that narrowing of the valve is much, much higher. And on the bottom, you can see that these kids had serial echocardiograms throughout their pathogenesis. They were born with it, and so we follow these kids every 2 years to see how their trajectory of them developing disease. So you'll see that there is one population of kids here that are good to go. They were born with a bicuspid aortic valve, but throughout their 20 years, they're just fine. They go out, right? They're doing exercise, they have no symptoms, and their stenosis isn't present. But we know they have a structural defect, but they do just fine. However, there are some kids that are born with a congenital, but they have this trajectory that just drives them to stenosis very, very early. And so why is it that some kids are just fine, but some kids do really poorly? So in order to understand that, we are trying to understand, we believe that these kids in the red box are the ones that calcify, okay? So again, you're born with it, but then you have a double whammy because you calcify, and that makes the cusps even stiffer. So these kids, and of course, we can't see if they're calcified, right? Because these are kids, these are living kids, so we're having echoes. They don't get CT scans on a regular basis, so we can't actually see if they're calcified. But our prediction is that they do poorly because they have calcium deposition on their cusps. These kids at the bottom don't develop calcification. So we went back to the literature a little bit and tried to understand what does calcification look like in people that are born with a bicuspid valve. And so if you were to take the aorta and fillet it out, you would see that normally you have these 3 cusps, the left, the right, and the non-coronary. If we were to look at what their calcification patterns would look like, it was published back in 1986, which I probably wasn't even born by that point. And so this is a very old paper, it's the only paper that we can find, but they showed that calcification happens in specific regions of the valve. It happens at the coaptation, so what you're seeing at the top is the same shape, right? So you can see that, for example, there would be, coaptation would be in this region here. And the coaptation is when the valve cusps open and close. This point where they touch each other is the coaptation point. So you get these calcification hotspots happening in the coaptation, the attachment region, which is where it joins the aorta, and then you get a combination of both. So this bottom left image, I want you to keep in your mind, so I'm going to keep bringing this back. So we have this free edge, the coaptation, the belly region, and the attachment region. But for some reason, the calcification doesn't happen over the entire cusp. It only happens in these regions. And so I'm going to show you a different view of the valve as we move forward in mice. And so I want to orientate you a little bit more. But now we're looking at the top of the valve, looking down. So when I show you these images, you'll see that this mouse, for example, has the 3 cusps. This one has the bicuspid. And so when I talk to you about the area of the coaptation, You might see it from that view, or you might see it from this view. Does that make sense? Yeah, I know that. And then I might talk about the attachment zone, and that's where it joins the aorta, because you're looking at now is the aorta, and these would be the attachment zones. So just to orientate you over, and I'll keep reminding you, but when I talk about these regions, that's what I'm going to show you. So the goal of our study is to really understand why 50% of kids that are born with this disease develop early onset. And there is a complete clinical need, because we treat all these kids the same. We follow them by echo. If their stenosis gets really bad, we do replacement. We take their valve out and we put in either a bioprosthetic or a mechanical. The kids outgrow them, right? Because kids are growing all the time. And so if you give them a bioprosthetic valve, it doesn't grow with the patient. If you give them a mechanical valve, they have to have antithrombotic therapy throughout their life. And yet we treat all these kids the same, but I've already shown you that 50% do just fine, and the rest of them have this trajectory. So we really want to try and understand and predict when these kids are born, which kids are going to stay in the green box, which kids are going to go into the red box. And that becomes really critical, because I say the interventions that are required for this, these kids have to keep coming in for reoperations every 10 years. Because if you put a bioprosthetic valve in, that calcifies, that becomes thrombotic, they outgrow it. And so this is a complete life cycle for these kids, and it's a big strain on them and their families. So our hypothesis is this calcific aortic stenosis is initiated because of the change in the anatomy of the bicuspid valve. And because of that, we have these changes in the biomechanical stress that these endothelial cells sense. And because of that, they activate these pathways that promote calcification. So I'm going to take you from the biomechanical indices to this, to the signaling pathways that are affected by that downstream. This is a long hypothesis, I don't recommend this in people's thesis. So to do that, we are taking kind of a multiple approach. It seems very systematic. It didn't happen this way, right? You always start with science and work backwards, but the idea is we're going to examine the structure of the valve. Is it bicuspid? What do the cusps look like? How stiff are the cusps? All these things will influence the blood flow and the mechanical indices, the function, the stress, and then the effects of that on gene expression, and then the readout. And to do that, we have, I'm very grateful, we have an outstanding patient population that are very willing to get involved in research. So we have over 400 patients that have bicuspid aortic valve disease that have had serial echoes. Some have the right-left fusion, some have the right-lung fusion, and then as our control, we have a population of kids that have a normal structure aortic valve, but they don't have a bicuspid. They were born with cardiomyopathy, but their valve function is just fine. In parallel with that, we have 2 mouse models of bicuspid aortic valve disease. One is a type 0, so the formation of those 2 cusps only, and then one of the models has a right-left fusion. And I'll keep reminding you of that. So I'm going to talk a little bit about the human work first, and I have to put the slide up because I'm in a biomedical engineering department. I am not an engineer, okay? So Josh is probably going to cringe at this and laugh at this. And my student wrote down very specific notes for me. I can find out more information if people need it, but I will tell you what I know, and it's not very much. So the idea here is that we took our patient population, and all of our patients get echo. Some get MRI. It's not very common. But for the patients that got MRI, we were able to find, this is just an example, but we have a 23-year-old who had a normal tricuspid aortic valve. We took their MRIs and we remodeled their structure. And this is really important because every single one of us, as you can imagine, our heart structure is completely different. So some people may have a wide aorta, a thin aorta, a heart, and so this anatomy is so important when it comes to understanding how the blood flow goes through that, because very slight changes in the anatomy significantly influences the speed of the blood flow, the direction of the blood flow, and all those factors will influence what the outcome is. So Hale took this and he reconstructed a 23-year-old normal patient and a patient that had bicuspid aortic valve disease in the absence of calcification. This patient had very, very mild stenosis. And the idea was that we didn't want to try and do a computational model on a patient that had full-on calcification, because that's just an added layer of complication. So we're trying to understand what does it look like before the calcification sets in. So we have our 2 subjects here, and then we did a reconstruction of the complete valve anatomy at the patient-specific level. That's all I know about what he did, okay? And then he took the phase contrast imaging from the MRI and he generated patient flow waveforms, which you can see here, so the flow over time. And then we used an in-house MATLAB, I say we, I didn't. Hale used an in-house MATLAB and he looked at the pressure gradients and the coronary flow waveforms for each individual patient. And through stuff I don't understand, we ended up with something like this for every single patient. And then we realized that we don't really know what the material properties is of a bicuspid aortic valve. So the published out there in the literature has been all on healthy aortic valves. And as you can imagine, the material property of the valve cusp is going to influence how the flow happens. So Hale did some biaxial testing on a human sample. Again, very fortunate I have a big tissue bank for congenital heart disease. So we collect iPSCs for all our patients, genetic sequencing, tissue, every single, any liquids that come from patients we bank. So we have this, and we're very grateful we had fresh tissue from a BAV patient. And so he took each of the cusps and did some biaxial testing on that as well. And so what you can see here is in the blue line is the normal patient, and then we took the non-coronary cusp from the patient, so that one is not fused, but then the right left leaflet are the ones that are fused together. So they form this raphe, and you can see it here. Can you see where they're fused? You can see there's like fibrous between where the two cusps are fused. That's like, it's like a collagen lump. And so it's really, really tight. And as you can see, when he is doing this, you can see when we do a longitudinal, so when we're stretching from pulling apart, you can see that this ends up having a very different situation here. And so the fused cusp is so much more stiffer than the non-fused cusp. And it's so much more stiffer than the normal. So already there is these material properties. So when we try to generate patient-specific FSIs for these patients, we actually put the material properties in as well as an added layer to get the answer to what we're looking at. So again, just to remind you of the anatomy, the structure, and where the calcification patterns happen. Again, this is just reminding you, because I'm going to show you now our patient-specific FSIs based on what Hale generated, and we're going to be looking at flow and time average wall shear stress on the fibrosa and the ventricularis side. So I gave this talk a few weeks ago at a non-engineering, and this was really for me, but I was trying to get in my mind, how do I describe what time average wall shear stress is? And the way I describe it is obviously the force that's coming through the riverbanks feel. So this is really what the endothelial cells are sensing, right? So as the blood comes through. And so what we did was, we did it for the fibrosis surface, which is away from blood flow, and the ventricularis surface. And so this is that patient-specific, we have the control on the top, and the BAV patient on the bottom as well. So the higher the red, the more wall shear stress is in those regions. And again, you can see the free edge, we try to divide up the free edge, coaptation, belly, and attachment. So, and then what we've done is we've quantitated it. It becomes very, very busy and very complicated, but we've quantitated this. And so really this just shows you what's higher and what's lower. But you're welcome, but I did a little summary for you at the bottom. So the idea really is that on the ventricularis surface, so the surface where the blood flow hits, as expected, those areas are so much higher than on the fibrosis surface. So you can see even in the control patients, there's blue pretty much all the way around. But again, this free edge here and the coaptation see a much higher degree of wall shear stress. And then when you have the bicuspid anatomy, that's just further exacerbated. So these patients, depending on their anatomy, this one patient that we did, when you have fusion between those two, you can see the wall shear stress is just so much higher in the free edge and in the coaptation region. And if you remember, there was calcification happened in the coaptation region. So it became very interesting to us that these patients really have calcification in the coaptation zone, but the wall shear stress is so much higher in these areas. So then we did the same for oscillatory shear stress as well. And again, I describe this as how the waves hit the shoreline, and that's the stress and the turbulence of the water. And then, so this again is the yellow is the highest, and the dark green is the lowest areas. And as expected, we can see already that, you know, in the control sample, the oscillatory shear stress is happening in here, but then again, in the wall, in the BAV, in the belly region specifically, the the oscillatory is much, much higher. And again here, when we look at the ventricularis surface, not so much is happening, but this is really happening on the fibrosa. And so if you think about how the cusps close, right, and then you've got that oscillatory happening on the top of that surface. So in the regions where you have the high wall shear stress, you have the low oscillatory shear index. So you have this kind of complementary patterns happening. And then finally we looked at von Mises stress. So really that's the sort of impact. I think like if you squeeze a balloon, that's kind of the pressure that you see from this stress. And we did that again. So when we look here at the BAV, we can see you have much higher von Mises stress in the belly region. And then when you look at the ventricularis surface, there's really this attachment zone really lights up. And so again, we have this high von Mises stress in the control, but that is further exacerbated in the BAV. So after many years of doing this in different patients, I summarized it in a table, but the idea is that you have high wall shear stress, high von Mises stress in the belly region attachment, but you have low oscillatory shear index. And so Hale did some models really to show the patient specific. And so now we have a pipeline where we can plug in all of our patients and find out how they look different, and we can relate that to their outcomes as well. And so the idea is one day, you bring your patients to clinic, you can use their anatomy that we can measure from their MRIs or echoes and use that to predict which patients are going to stay green and which patients are going to go red based on their flow structure interactions based on their anatomy. So now I'm going to move on a little bit to the mouse models that we have, and the idea is that we have two models. I'm not going to go into the genetics of the model, it's not really important, they're both published, but one generates this type 0 bicuspid aortic valve disease, and so the mice end up having this structure. These develop very mild aortic stenosis, and actually that's the same in the human population. But in our other model, they develop this type fusion, so they have fusion between their right and left. These mice develop very severe aortic stenosis at such a young age. So by 5 weeks of age, these mice are already stenotic. And it allows us to explore some of the questions we had in the human population, but using it in a mouse model. And so we took the same approach with structure, function, stress, and then we wanted to relay those maps I just showed you with gene expression to understand in those regions of high wall shear stress what's happened to the gene expression changes in those areas. And so starting with the structure function, what we did was Hale took the mice and we did 3D reconstruction to better understand what does the valve anatomy look like. So this is our control. This is a P1 neonate, so 1 day old pup, and he did a reconstruction. This is our control, 3 beautiful cusps, and then in our mouse model we have this is with the notch one. I indicate the top which mouse model. They have these 2 cusps at P1. And then by 5 weeks of age, we did another reconstruction, and they start to get a little bit thick at this point. And then at 10 weeks, you can see they are obviously still bicuspid, and then by 18 weeks, these cusps are thick, right? So you can see here that in our mouse model, and as you can imagine again, the structure of the valve cusp really influences how that biomechanical indices relay out. So the thicker the valve, we think that might further exacerbate disease because the valve is constantly changing. Now why is it changing? Is it because the flow is feeding back and it's thickening the valve? Don't know that, but you can see here the trajectory of what it looks like. And if we quantitate this, you can see that over time, this is our bicuspid valve, we have an increase in volume of the valve. So the mirror is nice because you can measure volume rather than thickness. And so by 10 to 18 weeks, these valves are significantly thicker than their genotype littermates. And then one thing we noticed is for those that work with mice, they never all behave the same. Even though they're in the cage, they have the same simulations in the cage. There's always some outliers, and we noticed there was some of the mice developed really, really thick valves. And so as we were going through this, we noticed that there were these outliers that were much sicker. We see that in our human population. Some kids do great, some kids don't. Same thing happens in the mice as it starts. So what we did after the structure, we looked at function. So we wanted to measure stenosis in these mice. And so I have a full-time sonographer, Carol, who was one of our pediatric sonographers. And then she got tired of kids crying and came to get mice biting her on our side of the street. So she does echoes 24/7 for all of us. And we noticed, so for those that don't read echoes, This is what a Doppler looks like, and it allows you to look at the blood flow direction. So red is the blood flow going in one direction, and blue is it when it comes back in the other direction. So in the control, you have this element of unidirectional, and so there's no mix of the colors, so this is going in the direction it should. But when you have the bicuspid aortic valve disease, the valve isn't fully open, and so you get this whole color spectrum. So this is an indication to us that the valve isn't functioning properly in these mice. And then we measured the peak velocity, which is very similar to the peak gradient in our human population. So the higher this number, the more stenosis they are, the more stenotic they are. And what you're looking at here is the mice at 5 weeks, 10 weeks, and 18 weeks. The gray bar is the ones with the bicuspid aortic valve disease. And even by 5 weeks, these mice are sick. And they get sicker, and they actually get sicker. So this is a nice model because their structure was bicuspid at birth, but they didn't really develop thickening valves until 10 to 18 weeks, but they are already stenotic by 5 weeks. So they're born with this anatomy, they're stenotic at 5 weeks, and then they get really thick, and then they get more stenotic, and so you've got this vicious cycle going on continuously. And again, we have these outliers of these mice that are really, really sick and stenotic. So then we try to correlate, is there a relationship between the valve thickness and the peak velocity? So the ones that are stenotic, were they the ones that had the thickest valves? And if we look here, this box here shows that these were the mice that really had the thickest valves and they had the more stenosis. So there's a relationship between having thick valves and a relationship between having stenotic valves, which kind of makes sense. If we took these 4 that stuck out on the spectrum, we did histology, we saw that there was calcification in that attachment zone. So they are thick, they're stenotic, they're bicuspid, and they have calcification as well. And so we think that's because they have this, this skew in the blood flow going through, and they develop calcification in this attachment zone. So, kind of putting all these pieces together. And so our next question was really thinking about the mechanical stress. So what do we know about structure function? How does that influence mechanical stress? And then that mechanical stress, how does it act on those endothelial cells to drive this calcification in these hotspot regions? So we are now doing the same thing that we've done in humans, but we're doing it in mice. And I wish I could show you the data, but we are still trying to get this to go. Mice are much smaller, but their anatomies are also very different. So again, we're doing this in a mouse-specific simulation as well. And so to look at the gene expression, we've been doing spatial transcriptomics, and for those that are not aware, what this allows you to do is you can take a tissue section of any of your favorite organs, and basically on the tissue section, you put it onto a slide, and the slide has the entire genome on there. So when you then put your tissue section on top, you can look at differential gene expression changes in the regions, like spatially, right? So when you do single cell, you collect all the cells, you do your analysis, but you don't know where those cells came from in that tissue. This allows you to do that. So you can take a section, for example, of your favorite tissue, your favorite organ, and you can then look at the gene expressions in that situation. And so, just to give you an idea, the idea is we're trying to look at mechanical stress, and we're doing this now in mice in a 3D way. So those colored maps I showed you earlier in the humans, this is what we're generating in the mice. And so we can cut through those and take tissue sections and look at how these different patterns of mechanical stress relate to gene expression. So each one of these dots here is a genome, and it will allow us to test gene expression changes between dots. Okay, so we can test it in the control, and we can test it in the bicuspid. So this is one of our mouse models, and so again, each spot is the genome, and what it does is it colors, the colored dots relate to how similar the molecular chain or the molecular profiles are within the heart section. So what you're looking at now is a cross-section of the mouse heart, and based on the molecular similarities, it identified the aortic valve in pink. So they knew it was a valve because it expresses its own molecular profile. So then we compared what genes are changing in the pink-only cells between our control and our bicuspid mouse model. And there were 102 genes that went up in our bicuspid model, and the 546 genes that went down. These are 18-week-old mice, and they haven't calcified yet. So they're bicuspid, but they're not calcified. So this allows us to look at early changes prior to the calcification happening. And very interestingly, we saw calcification genes that were changed, even though there wasn't calcification yet. It was already changing at the molecular level, so we think this is like a pre-calcific change before we get to that point. This mouse model is the mild one, so again, there was no calcification happening, but we did see upregulation of this SPP1, which is a glycoprotein involved in bone mineralization, in those attachment regions and the coaptation regions. So this is kind of an indicator that there is already pre-calcific changes happening in these mice in the absence of calcification. We then repeated this using the more severe mouse model. Again, we picked up the molecular profiles of the aortic valve, and we looked at that. There were 92 genes that went up, 182 that went down. We also saw an increase in SPP1 in the attachment zones, and these mice do calcify. So this calcific region here in red is alizarin red, it picks up calcium. You can see that there was the higher gene expression levels of this pre-calcific marker. And then when we looked at cross-section, this is the attachment zone. And purple here is the calcification, true hardcore calcification happening. You can already see that these cells, can you see like how they're big cells with big cytoplasmic, they're like chondrocytes, osteocytes. And then when we overlap that with some of the genes that we pulled out from the spatial, you can see there's an overlap between calcification and these pre-calcific areas. And again, this was the attachment zone, which I've shown you already was a hot spot. And where we showed from the human data, there was high wall shear stress happening in those areas. And again, this is the von Mises stress. So we had low von Mises stress, high wall shear stress in this attachment zone. And then we now think then that there is something going on between the bicuspid aspect that gives you a patient-specific changes in the wall shear stress or the biomechanical indices that then transmits something. We don't know what it is, but there is a bone-like program happening in these cells. So how do we get from the endothelial cells at the top receiving the signal from the flow in the wall shear stress to these cells that are turning into bone? There's a missing factor. So we went back to our spatial transcriptomics and we pulled out ATP-associated processes that was actually similar in both models of BAV. When we looked at all the differential expressed genes, we plugged it into gene ontology, and this ATP biosynthesis changes. So there is some suggestion that there is something going on in the endothelial cells related to ATP biosynthesis. And when you have this altered shear patterning across the cells, they respond very differently. So one of those genes that changed was P2RX4, and P2RX4 is a receptor for ATP. So we pulled this gene out, and it was upregulated 1.6-fold in our spatial. And so, for example, this is one of our mouse models. The P2RX4 by spatial increased, so the red is the higher, in the attachment zones and the coaptation zones. And then when we actually validated that, you can see in red, you can see where P2RX4 is in the attachment zone and the coaptation. So there's definitely an increase in the ATP receptor in bicuspid aortic valve disease. So maybe there's some kind of altered metabolic signaling going on that's the missing link. And this actually wasn't a surprise because this has been described in vascular endothelial cells. So in vascular endothelial cells that see shear stress, they increase their levels of ATP in response to shear stress. So we went back and we exposed our cultured valve endothelial cells to the shear stress that Hale pulled out from our human population. So now we know exactly what the shear stress level is that those cells see. We collected the supernatant, so the endothelial cells will spit out the ATP, and the idea is we then put that onto the interstitial cells and see if we can get them to undergo calcification. So to do that, we have the Abili system, and we expose the endothelial cells to patient-specific BAV shear stress. And just in proof of principle, we showed that whether you expose them to oscillatory or laminar at different levels, we can actually increase the amount of ATP being produced. So ATP is in red, and you can see that the 5 dynes increases more ATP than it does at 20 dynes. And this work being done by McKenna. And so she then took that supernatant, or she added ATP and onto our valve interstitial cells to see if they would calcify in response to ATP. And the purple dots is alkaline phosphatase, which is a marker of bone in calcification. And so when you just add ATP to these cells, they undergo calcification. And then she did it in the presence of the P2RX4 inhibitor and showed that ATP can drive calcification via the P2RX4, and that was quantified. And so really thinking about how do we use antagonism of this ATP receptor to treat this. So we've been doing a study in our mouse model where we know these mice get stenotic, they get these pre-calcified lesions where the P2RX4 is with the bone markers, and they develop calcification at these different time points. So we have a two-prong approach. One is we're taking our mice and we're injecting them with this P2RX4 inhibitor at 5 weeks, and the idea is that we stop them getting calcification. So before they show this, we stop them getting. The other way is that when they get to the calcified point, then we treat them and see if we stop it getting any worse. And so for the prevention study, we have injected mice with either the drug or the vehicle. This is stenosis. So in our vehicle treated, if you have bicuspid valve disease, you get stenosis. But in the few that we've tried, our drug doesn't seem to prevent that stenosis. And if we look at the histology, P2RX4 in red is pretty much decreased in our bicuspid mice because we're inhibiting it. But then if we look at the markers of calcification, in this case SPARC, that's also decreased. So it looks like in the prevention study, if we inject our mice at 5 weeks, we are able to prevent the stenosis happening, and we're also able to prevent those pre-calcific changes happening in the tissue cells. So in summary of everything, I hope I've tried to show you that we have these regionalizations of altered mechanical stress on the valve endothelial cells. And then when that threshold reaches a certain point, it increases ATP release from these endothelial cells that acts on the receptor that we've shown in the interstitial cells and drives them to this calcification program. And again, this is what the regionalization looks like. So that's all I've got. Really, this has been work done by 2 graduate students. So Hale has driven everything, to be fair, and then McKenna, who is a new graduate student, she's going to pick up the story of the ATP and continue this on. We're very fortunate to have John Adisa in my group, who also has been helping with computational and funding as well. So thank you. Take any questions. Yeah.
That was awesome. Thank you for that. You might not be an engineer, but I am not a biologist. So one quick clarification question. Your mouse models.
Yeah.
The NFAT-C1 CRE versus the NFAT-C1-EN CRE. What does the EN stand for?
Yeah, so the EN is the enhancer CRE, and so it recombines in a slightly different valve endothelial cell population. So the full, without the enhancer, recombines in all the endothelial cells and the interstitial cells. So in that model, we're losing the XOC5 in all the valve cells. In the other model, we're losing Notch1 in just the endothelial cells.
Okay.
Yeah, and it's very difficult to compare the two because the efficiencies are very different. It's a great question. For now, I think accept that they are models for these two diseases. But yeah, it's a great question. Yeah.
And then I have one more. On your hemodynamic models,
Yeah.
when you're looking at the aortic shear versus the ventricular shear,
Yeah.
how much does the position of the coronary ostia affect what you're seeing? Because from what I saw, it looked like your patient-specific models had a little bit of high takeoff.
Yeah, the biggest thing is the distance of the valve height to where the coronaries hit. That's been our biggest influencer of changes in the patterning.
And then do you see like BAV patients tend to have higher or lower coronary ostia compared to tricuspid?
I don't know. Yeah. And it's interesting because if you have a tricuspid normal valve, the non-coronary cusp seems to have a very different calcification rate than the two that have the coronary. So certainly the coronary flow influences the calcification for sure. Yeah. And of course, when you start calcifying, that gets narrower as well. So you're just,
Right.
this,
Yeah.
cycle constant. Yeah.
Have you identified any co-founding variables or like neglected factors that you think could be affecting the prevalence of cardiovascular disease?
From the patient-specific thing, I think the valve height for sure. When you look at the valve-specific anatomy, that's been one thing. And the other thing is the material properties. We're really finding that, so what I didn't show you today, because I would have butchered it completely, but what Hale did in addition to the BAV patient, he took the normal patient and artificially fused them in the program. And then he changed one with the material properties, he changed one with the specific boundary conditions to try and see which ones really influenced that. And the biggest thing that changed that was the material properties. So I think, and that's hard to test in patients, right? Nobody's going to give up there. But I think it's one thing to understand is if we can measure material properties by imaging, because if we can look at how on echo or MRI, if we can measure kind of stiffening indirectly, that would be a variable that might suggest, okay, you're going to be influenced because of the speed or the rate at which your valve opens and closes based on material properties.
Thank you for the talk, it was amazing. So I have a question, since like notch is a very important signaling pathway to
Yeah.
the development of the valve.
Yeah.
So do you induce the knockout of this molecule at a certain point, or like from
Great
the beginning?
question. Yeah, from the beginning actually. Well, these are actually these are hex for the notch model, so they're only losing half of their notch. They anatomically, they seem to develop valve development just fine. I'm not sure how much has really been done, but the BAV happens. So when I say that, the endocardial cushion development happens fine. But then, because we're doing it from birth, and these are congenital, during their embryogenesis, they're born with this, right? So something is happening during embryonic development. It's a great question. Actually, if you delete all of notch, then you're in trouble, right? And notch has been shown to play a role in BAV in the human population. So you're completely right. The notch depletion significantly affects the valve development, post-endocardial cushion development, leading to this fusion somehow. And we don't know if these mice have 3 cusps and then they fuse, or they just never form. I think it's probably the latter, that they just never form. But yeah, great question.
Or the entrance is 100%, right?
Yeah, so the notch one line, we have about, every single aortic valve is plastic. I would say about 90% have BAV. For the other line, it was horrible. We had like 5% BAV, and that's why we stopped working on it, because poor Hale was just, yeah, it was a lot. And so the notch line is a beautiful line to work with. Yeah, they all get it. It's really sweet.
If I'm not mistaken, you mentioned that there are like several metabolic genes that could be studied. I was just curious why specifically P2RS4 out of all of them?
Yeah, honestly, it was kind of one of those things where, when you do a non-biased approach, but then you ignore the things you don't want to work on. So we pulled it up, and actually there was a link between P2RX4 and so we did it so many times, and actually for the I didn't go into it, for the spatial, we did mice that had the same genotype but had low stenosis, high stenosis, thick valves, and so we've got a variability to see if stenosis might influence gene expression. And the only gene that was significant across all those conditions, well, it's not the only gene, but one of the few genes was P2RX4. And so we did it, and then there's a link with P2RX4 and bone development actually. So P2RX4 plays a role in bone development in the skeletal system. And so putting those few things together, it just became one of our favorites. Yeah.
For your in vitro studies that you did in the ATP and the culture media that you
Yeah.
put on the cells, did you just take all of the culture media
Yeah.
and was that like supplemented with ATP? I think
Yeah, we've done both. So what I showed you there was just ATP, no media from the cells. But we are doing that. The problem is ATP is so short-lived, right? So we're having to put anti-ATP degradation enzymes in to stop it from being hydrolyzed. And also they produce so much. The volume of the immune system is like 20 mils. So when these cells spit out the ATP, it's in this huge volume. Trying to concentrate it down to see if we can find a way of trying to get it to do that. But yeah, good question. Yeah.
I have a very basic question. This is not my area, but so DNA is normally very tight surrounding these histones.
Yeah.
And in order to do that, this RNA polymerase cut in the right position, you have to release the force. People, metallization,
Yeah.
population,
Yeah.
forces, mechanical forces may create that release. Do you see a connection between this stress and changes in the organization of the DNA that could create the situation in which this polymerase comes at the wrong position?
I don't know the answer at all, but I think one thing that has been shown is that the shear stress influences the cytoskeleton of the cell. It can elongate the nucleus, it can have many different changes for the active cytoskeleton, the microtubules. So it wouldn't surprise me if there was an indirect way in which gene transcription is being modulated by shear stress based on cytoskeleton or the nucleus deformations. Yeah, I don't know if anybody's looked.
All right. Let's thank Dr. Lincoln again. Have a good spring break. See everybody in a couple weeks.
Yeah.